Hi, everyone. It's scary to be up here. Yeah, so thank you, Lluis, and all of you for organizing. It's nice to be in Paris. So I'm going to talk a little bit about Denisovan ancestry and Neanderthal ancestry. And so before I do that, I want to acknowledge the people who did the work that I'm going to show you. And before I forget, I want to let you know that I'm looking for postdocs in case someone is interested. And what I'm going to talk to you about today is something that was actually just recently published. So if you want to read the article. Okay, so I'll start with an introduction, and I'll say that I think we are all in agreement that human origins are in Africa. But since then, humans expanded within Africa and also out of Africa. And when they expanded into Eurasia, they encountered Neanderthals. And we know from the archaeological data that they overlapped both in time and space, raising the possibility that maybe they admixed in the past. But of course, that was something that couldn't be done using archaeological data alone. And so in 2010, the first in a project spearheaded by Svante Paavo, the first draft of the Neanderthal genome was sequenced. And so that really enabled scientists to direct this Neanderthal genome to modern human genomes, and folks were able to identify Neanderthal tracks. And so this allowed both to study the evolutionary relationship between Neanderthals and modern humans, but also to provide definite genetic evidence that humans and Neanderthals had interbred. And so I was actually a postdoc in Berkeley when this happened, and I was not associated with the study, but I was surrounded by people who were. And I remember having lots of lively discussions about what was going to be the utility and the significance of having one Neanderthal genome. And some people thought that it was not going to be that significant because at most we would only be able to use it to identify human-specific changes, since we used to use the chimp before that. But looking back now and seeing the talks from today and other talks, I think I can say that it's been pretty significant, as it opened up new lines of research, not just for me, but for others, and that have only grown in the last 10 years. But it also has been significant for the study of human evolution. Another thing that has changed is the narrative of Neanderthals. So we've known about Neanderthals since the 1800s, and this is an early depiction from the early 1900s. And so they used to be depicted as being different from us. People used to think of them as being inferior or less intelligent. But of course, having access to those genomes has changed not only how they're depicted now, they look more like us, even though we actually don't know what they look like, but that the narrative has definitely changed. Okay, and so one interesting, I think, thing about Neanderthals is that most of the archaeological sites happen to be in Western Eurasia. And so looking at that, one might think that perhaps the populations that have the most Neanderthal ancestry are populations in Europe. But when people have looked at what individuals actually have the most Neanderthal ancestry, it's populations in East Asia, in South Asia, and the Americas. And so Neanderthals are not the only population that interbred with modern humans. So this is a picture from the Altai Mountains in Siberia, which is present-day Russia. And we know that multiple human groups have lived in this space because Denisova cave is here, and that cave has been a really rich source of genetic material from different archaic humans. And just to show you some of the fossils that have been excavated, we know there's fossil remains from humans, Neanderthals, and Denisovans. And of course, Denisovans, I think, are interesting because we didn't know they existed until roughly 10 years ago. And that's simply because there was a whole genome that was sequenced from a small finger bone, and comparisons of that genome to other Neanderthal genomes and modern human genomes led folks to realize that it was not a Neanderthal, not a modern human, it was a Denisovan. And so we introgressed with Denisovans as well. And when you ask what populations harbor the most Denisovan ancestry, again, it's not populations near the Denisova Cay, which is in Siberia. It's populations in Southeast Asia, especially the region of Oceania. And perhaps due to, and there's lots of hypotheses as to why that is. Okay, and so just to summarize, in the last decade, I would say we have spent a lot of time trying to quantify what are the levels of archaic ancestry in different individuals from different human populations. And so it's roughly between 0.1% and 7%, which may seem small in quantity. But studies have shown, including mine, that some of this genetic variation that enter modern human populations have been really influential in both facilitating or perhaps accelerating human adaptations. And so one clear example of that is high altitude adaptation in Tibetans, where we know that Tibetans are not just acclimated to live at high altitude, but they are genetically adapted. And so that genetic variation that's beneficial did not originate in Tibetans, but it originated in Denisovans. And there are other examples of that, genes in skin pigmentation, immunity, and metabolism. And so just to summarize, we know that we introgress with archaic humans. We know that some of this introgress variation was targeted by natural selection. And something I didn't say, but we also know, is that most of this variation is probably deleterious because there are introgression deserts and so on. And so the motivation for what I'm going to show you right now is we know that we introgressed with archaic humans in the past, but since then, humans have continued to evolve, and so has that variation in those populations. And so one thing that we wanted to do is look at that variation in different human populations and see what is the distribution of this variation in different populations. And one thing we were interested in is that because there's only a handful of archaic genomes, there's only one Denisovan genome, for instance, and a few Neanderthal genomes, but we have a lot of sequences from modern human genomes, each of which have some archaic ancestry that survives in them. And so the idea was to understand whether we could actually use, could we actually use the surviving sequences in modern humans to perhaps build pseudogenomes of archaic humans that we can then use? And so this led us to a very simple question, which was, how much of the archaic genome can we recover as we increase the sample size? Okay? And so imagine that you have these individuals here. So you have 4 individuals. And so here, let's imagine that they have a small genome that's 10 kb. And here the gray boxes could be introgressed segments from archaic humans. And so for this individual, for instance, that individual has 4 kb out of 10 kb of Neanderthal, let's say. This individual has 4 kb, this one has 6, and this one has 5. And we can also think about a summary for the group of individuals where we can create a one-dimensional summary of how much we can recover from all the 4 individuals. And if we do that, if you pooled all of them, you get 7 kb. So you get a little bit more in the sample than you do in each individual. Now, depending on what the distribution of these segments is, you could have something like this, where this individual has 3 kb, this one has 3 kb, this one has 3 kb, and this one has 3 kb. But when you pull them together, you recover a whole genome. Okay, so essentially what we wanted to do is use information from these tracks to be able to answer that question, how much of the archaic genome can we recover as a function of the sample size? And so the first measure we did is we just counted archaic alleles, which are defined by two features, meaning that they are rare or absent in Africa, and that the allele is rare or absent in Africa, but it's present in a recipient population. So you can think of these as being alleles that are within these introgress tracts. There could be, if we just count these variants, there could be cases where they actually don't lie within a tract, but we think that they're there either due to incomplete lineage sorting, but presumably those will be there at similar rates in different human populations. Okay, and so if we just count archaic alleles in individuals in populations, this is what we have. So here, every point is just to count for an individual in either an East Asian population, a European population, in the Americas, or in South Asia. This is data from the 1000 Genomes Project. And these are counting Neanderthal alleles. And what you see here is what we expect, that East Asians have a lot of Neanderthal, more than Europeans, and more than South Asians, and more than the Americas. Okay, that's what we would expect. If you look at the Denisovans, you see that East Asians have more than South Asian, well, more than Europeans for sure, and a little bit more than South Asian populations. So at the individual level, we see what we expect. Now, the question was, what do we see when we increase the sample size? Well, when we increase the sample size, what you find is that you start recovering more Neanderthal, not from East Asian populations, who have the most at the individual level, but from a sample of South Asian populations. If we look at the Denisovan variation, you see again that you recover the most Denisovan from a South Asian population than from an East Asian population, despite the fact that at the individual level, East Asian populations have the most. Okay, and so in population genetics, we know that most of the variation is shared in humans. And so one thing that we wanted to ask is how is archaic variation shared between amongst human populations? And when you look at Neanderthal alleles, you find that you see what we expect, that most of it is shared between populations. Every pair of populations, there's large, or if it's in one population, it's likely to be present in another population. But when you look at the Denisovan alleles, most of it is not shared, most of it is private. So, for instance, South Asian populations, oh, you can see that, well, it's 44.7% in the South Asian populations. 44% of the total number of Denisovan alleles are private to South Asian populations, and 27% East Asian populations. And a lot of it is not shared between these two populations. And so one possibility is that we know that there's been multiple waves of introgression from distinct Denisovan populations, and perhaps these contributed differently or uniquely to either different geographical locations. Okay, so another thing we wanted to look at was the distribution of archaic variation. And in particular, we wanted to look at variation that happened outside of Africa. So when humans expanded into Eurasia, they could accumulate mutations two ways. One was by de novo mutation, and another one was through introgression. And so the idea was to see whether that new genetic variation was whether human demography potentially had impacted these two sets of variants differently. And so the set of variants that was due to de novo mutations, we call those non-archaic variants, and the ones that were due to introgression, we call them archaic variants. And we classify them into low frequency, in here that means less than 20%, or high frequency, bigger than 20%. And so to look at this, we looked at this ratio of archaic common, archaic rare, over non-archaic common, non-archaic rare. And the idea is that if demography has impacted archaic variation in the same way as other variation that happen outside of Africa, this ratio would be 1. But if it's not 1, then it would be that would mean that either the archaic variations would have higher frequency than the non-archaic variation, or vice versa. And so when we look at that, so on the x-axis we have the denominator, and on the y-axis we have the numerator. And what we can see here is that in most populations, the archaic variation tends to have higher frequencies, but in the South Asian populations, it tends to be rare. And so what this means is that South Asian populations have more, we recover more of the archaic genome from South Asian populations, but the variants themselves happen to be at lower frequencies. Okay. And so another question that has received a lot of attention is, why do East Asian populations have more Neanderthal ancestry than Europeans? Okay, so here I'm showing you again, this is the levels of archaic ancestry at the individual level in East Asians, and this is in Europeans. And so several explanations have been proposed. For instance, one is that maybe there was more Neanderthal introgression events into ancestral populations of East Asians. Another one is that maybe because East Asian populations have a lower effective population size than Europeans, natural selection was less effective at removing these archaic variants. Another one is that there was a dilution in Europeans, meaning that Europeans admixed with modern human population that did not introgress with or did not have any Neanderthal ancestry. And another one is that Africans could also have Neanderthal ancestry, and so if we use them as a control, you mask some of that Neanderthal ancestry in Europeans. And more recently, by Moy, who's in the audience, that there are differences in generation time. So recombination rate is larger in one than the other. All right. And so when we look at our results, we see actually that when you increase the sample size, we recover more from Europeans than from East Asians. So even though East Asians have more Neanderthal ancestry than Europeans at the individual level, you recover more from Europeans as you increase the sample size. And so one explanation could be that the introgress tracts tend to be more homozygous in East Asian populations. And so, for instance, if you measure homozygosity of tracts in East Asian population, it tends to be higher than Europeans. And so what that means is that there's more sharing of Neanderthal variants in East Asian populations than in European populations. Okay, and so what we wanted to do was look at whether or not one of these explanations could lead to this pattern. And so what I'm showing you here is sample size on the x-axis and the East Asian European coverage ratio. So how much basically the amount you see in East Asians over the amount you see in Europeans. And so at a sample of size 1, you see that it's bigger than 1, but it decays as the sample size increases. And you can see this either counting SNPs, or you can see this if you use different introgression maps from that are available, including some from the folks here in the audience. Okay, and so one thing we wanted to see is whether 2 pulses of introgression could explain this pattern. And so for that, we needed to generate some genomes, and we needed to assume a demographic model. And so we used this demographic model. So this is the present. And we use parameters from, we call it the Simon-Gravel model for the European, African, and East Asian parameters. And then we added this Neanderthal population. And we only vary 2 things. we varied the proportion of introgression and the number of pulses. So you could either have a model with 1 pulse of introgression in the ancestral population of Europeans and East Asians, or 2 pulses of introgression where the second pulse was in East Asian populations. And so the idea was to ask, if we varied the pulse and the number, which of these models could lead to the pattern that we observe? And so it turns out that none of these really are a good fit to the data. So the black line here is what we observe, and these other lines are the lines of the best fit models. And it turns out that whether you use a 1 pulse or a 2 pulse model, you can generate this pattern of more at the individual level and less as sample size increases, suggesting, I think, that demography has an effect on that. But as you can see here, none of these are a good fit to the data, suggesting that we perhaps are missing some. We're missing some aspects of the model that our model is not capturing. Okay, and so just to conclude here, we find that East Asian individuals have, at the individual level, more Neanderthal than all the populations we considered. We get more Neanderthal, but even though they have more at the individual level, as we increase the sample size, we have more Neanderthal from a set of Europeans than a set of East Asians. But we actually recovered the most from a set of South Asian individuals. And in models we also show that in models with 1 or 2 pulses that neither one or two pulses of introgression in a model can actually explain our observed ratio of East Asian over European as a function of sample size. And we also find that even while we recovered the most from South Asian populations, from Denisovan, we recover more Neanderthal and more Denisovan from South Asian populations. A lot of that genetic variation is it's rare. Okay, and with that, I want to thank you, and I want to thank my lab. And if I have several postdoc positions open, so if you're interested, talk to me. Thank you.